Huh? Who are you? And where am I?
Hi, I am Mune. You're in the Bioland. I'll assist you through here. So let's go to the DNA, Recombinant DNA Technology then.
Okay, but what is recombinant DNA technology?
I will explain when we arrive, as soon as we arrive. This is the definition. I am in search of the definition. So, the definition is a method in which DNA molecules meet in vitro with segments from different sources or by combining genes from different organisms.
Hey guys, ooh, newcomers.
Hi, I'm Irfan.
I know you. I'm Suha.
Suha, do you want to show him around your department?
Of course I will. I'm so excited. Let's go. I show you around.
I have a book too, so I will.
You not follow us? Okay.
Bye.
Bye.
There are 5 tools used in recombinant DNA technology. The first one is target DNA. The second one is restriction enzyme. The third one is DNA cloning vector. The fourth one, DNA ligase. And the last one is host cell. Target DNA. Target DNA is a sequence of DNA containing gene of interest. For example, gene encode for protein which can dissolve blood clots used in heart attack therapy. Second one is restriction enzyme. Enzyme which cuts DNA molecules at a restriction site and produce short restriction fragments. Restriction site is the short base pair sequence of 4 to 8 base pair recognized by a particular restriction enzyme. Restriction site contain palindromic, where the same sequence of bases is found on both DNA strands but in opposite directions. There are 2 types of cuts, which is sticky end and blunt end. The example of restriction enzyme is EcoR1. EcoR1 is produced by E. coli. It is recognized the base sequence of GATTC. It makes staggered cuts within the sequence. E. coli produces sticky ends. The second example of restriction enzyme is SMO1. SMO1 is produced by Serratia mercescens. It is recognized the base sequence of CCCGGG. The cut is made between the adjacent C and G. The cut straight across the double helix and produce blunt ends.
Please have a seat.
Thank you.
Oh my god, is it fine?
Yes, it's him. I was showing him around our department and the thought came about target DNA and precision enzyme.
So, what's your name here?
Actually, I want to ask you a question.
No.
Please?
No, I was busy lady.
No, please?
No.
Can you continue showing me around? Because,
It's make me fat.
candy? You love sweet.
Also make me fat.
Energy bar.
Oh my god, last time I ate it.
Yes.
Very good.
Hello there.
Shut up. Energy bar. What are your energy bar? Lots of it.
Okay, sure, fine. Okay, let me show you around. First, I will show you about DNA cloning vector. Cloning vector is a DNA molecule that can carry foreign DNA into a host cell and replicate there. This DNA cloning vector has their own characteristic, which is able to accept foreign DNA in multiple cloning sites, then able to replicate freely in the host cell because it has origin of replication initiation, or we call it as ORI. Next, possess selectable genetic marker which is resistant to antibiotics. There are some DNA cloning vector that we can use. As an example, we can use plasmid. Plasmid is a DNA molecule that can carry foreign DNA into a host cell and replicate there. The capacity to carry insertion sites is 1 until 10 kilobase. Next, we can use bacteriophage. Bacteriophage is a virus that infects bacteria. The capacity to carry insertion size is 8 until 20 kilobase. Next, we can use cosmid, which is a hybrid vector, a combination of plasmid and cos gene of phage DNA from bacteriophage. The cloning capacity is 35 until 50 kilobase pair. Last but not least, we can use YAC. YAC is a fragment of DNA from the human genome inserted into a yeast cell. YAC has the biggest capacity, which is 100 until 1000 kilobase pair. Next, I will explain to you about host cell. Host cell is a cell that can receive a recombinant DNA and allows the replication of recombinant DNA. Host cell should be able to receive recombinant DNA through transformation process. Next, it should be able to maintain the structure of recombinant DNA from one generation to another. And also, it should be able to amplify the gene product from the recombinant DNA. Usually, we will use bacteria because bacteria DNA can be easily isolated from or introduced into a bacterium. Last from my department is modifying enzyme. We're going to talk about DNA ligase. DNA ligase join the foreign DNA fragments containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester bond to form recombinant DNA. Next, Taq polymerase. Taq polymerase used in polymerase chain reaction, or we call it as PCR. Taq polymerase used to replace the DNA polymerase because of the temperature condition of PCR. It also used to add nucleotides to the third end of primer in order to produce new DNA strand.
What are you doing?
You prefer, right?
Yeah.
Oh, everyone in Violet know you. Oh my gosh. By the way, I'm doing screening. Blue screening.
Blue screening?
Blue screening is a step in gene cloning.
Oh, really?
Before I show you how to do the blue-white screening, I will show you there's 5 steps in gene cloning. First one, isolation of gene. Isolation of gene is plasmid from bacteria cell is isolated. Gene of interest is isolated from DNA to the donor cell. Secondly, cleavage or cut. Cleavage or cut is when DNA donor, for example human, and plasmid bacterial is cleavage using a compatible restriction enzyme. Make a single cut within the ligase gene site of cloning and providing complementary sticky end. The third one, insertion. Insertion is DNA fragment gene of interest is inserted into the plasmid. It's joined together by DNA ligase, then forming a recombinant DNA. Fourth, transformation and amplification. Transformation and amplification is the recombinant DNA then insert into the bacteria through the transformation process. As the bacteria reproduce, the recombinant DNA is also replicate, clone. Lastly, screening. To identify the bacteria is done containing recombinant DNA, blue-white screening is done. Plate of bacteria on agar containing antibiotic, implicit and sugar agar. It's incubate until colonized grow. Screening process is done using a blue-white screening technique to identify bacteria that carries the recombinant DNA. First, plate the bacteria in agar containing antibiotic, amplicin, and agar. There's two possibilities. The first one is non-resistant to antibiotic amplicillin and the resistant to the antibiotic amplicillin. For the non-resistant to the antibiotic amplicillin, the bacteria will not grow. For the resistant to the antibiotic amplicillin, the bacteria reproduce and form colonies. There is also two possibility. There is one colonies without recombinant plasmid will be blue. Meanwhile, colonies with recombinant plasmid will be white. The recombinant plasmid that blue is intact with like Z. They also can produce beta-galactose and hydrolyze X-gal. The colonies with recombinant DNA plasmid do not contain intact like Z. They cannot produce beta-galactose and cannot hydrolyze X-gal.
From the diagram, the figure showing compatibility of cat plasmid with foreign DNA and recombinant plasmid. Plasmid DNA is isolated and cut with the same restriction enzyme that used to cut human gene. Both plasmid and isolated human gene have no complementary or same sticky end. The both digested plasmid and human gene fragments are mixed. Foreign DNA is inserted into the cut plasmid. Hydrogen bonds are formed between complementary sticky ends. DNA ligase is added to join both DNA by forming phosphodiester bonds. And lastly, plasmid containing additional human gene is produced.
With that concludes all the recombinant DNA technology that we can show you.
Oh, great!
So, what did you learn from the tour?
What I've learned in this tour, there are 5 tools used in the recombinant DNA technology. The first one is the target DNA, and the second one is the registration enzyme. Then there are the DNA cloning vector. There's also the host cell and the modifying enzymes. Then there are 5 steps in the gene cloning method. The first step is the isolation of gene, and then the cut or cleave method, and then the insertion. After that, there are transformation and the amplification. The last step is the screening, known as the blue-white screening.
I think you got it all. So, let's meet the other before you leave.
Okay.
Hey you guys! So, it's time to say goodbye to Ifan. Ifan? You alright?
Wake up!
What happened?